And now we come to the first speaker. As I said, Shawn Lehman is a professor at the Department of Anthropology at the University of Toronto, and his main interests are lying in the evolutionary ecology, in landscape ecology of lemurs and trees, and in conservation biogeography of primates. And he has been for many, many years active in Madagascar. Before that, he worked in Guyana, and maybe he will be talking about both regions of the world. We will see that in a minute. But I'm very happy to have him here. He's an expert on Malagasy forests and lemurs, and he will talk on fragmentation effects. And now maybe you want to share your presentation, Shawn, to get started.
There we go. Can everyone see that all right?
Yes.
Okay. Oh, one moment. He wants to start at the wrong spot. One moment, please. There we go.
You have to share again. At least I can't see it yet.
Okay, one moment, sorry.
Yes, that works.
There we go. Does that work?
See the presentation, it's still not yet.
All right, sorry, it's saying it's not at the front window. One moment. It's always something. All right, we'll try this. Now it should work.
This looks good.
Okay, great.
Yes.
Well, thank you very much for the introduction, and thank you for having me today. It's a real pleasure. I guess I'm your overseas guest, along with Bertrand and some other attendees from continental Africa. I've been asked today, and I'm very happy today, to talk about forest fragmentation effects. It's been a considerable interest to me for quite some time in kind of a strange way. I started working in South America many, many years ago. I won't say how long. And this is what it was like when I entered into the forest. This is Kaitru Falls National Park in Guyana in South America. And it is one of the few countries in the world, in the tropics, where the forest just goes forever. It is a continuous canopy. Approximately 76% of the forest is still intact in Guyana. And I spent many years wandering through this forest, sometimes not seeing direct sunlight for many days. And despite the continuous nature of the forest, the complete beautiful canopy and the amazing sights that you see, what always struck me was the depauperate mammal community. They were there. Ocelots, jaguars, giant river otters, caiman, giant anteaters, but it was incredibly rare to see them. You would go days and see one large mammal. And yet when you traveled to a more fragmented landscape, such as this along the Manaus River in Amazonia, it was very common to see many different kinds of animals, an incredible diversity of domestic and wild animals, including the most dangerous of all domesticated or wild animal, which is human beings. But it always struck me as quite amazing is that in this so-called fragmented system, you saw such remarkable diversity. And that really piqued my interest in further studying fragment forest effects. Now, before we get into it, I just thought we'd sort of just very quickly go through some very straightforward operational definitions of what I'm going to be referring to. And fragmentation, quite in its simplest form, is just breaking up a continuous forest into smaller parts. And whatever those parts are, it doesn't matter as long as it's still some kind of an organism, plants, it's got animals of some kind. And that's juxtaposed with forest loss, which is both the reduction or size loss of a patch. And a patch in landscape ecology is just a habitat. In primatology, we tend to refer them as fragments because our animals are almost always found in forests. But in ecology, we refer to this as a patch. Now, dissection is a different kind of fragmentation process, and it's where there's a subdivision of a once continuous landscape. And so down at the bottom here, I've drawn a diagonal line for a continuous forest. And that could be created by a road, for example, but it could also be created by a path. It could be created by a river. It could be created by almost anything you can think of, a power transmission line. Anything that creates a disjunction in the continuity of a landscape habitat. Finally, there's perforations. These are holes punched in a landscape. And we tend to think of these as sort of the classic indigenous people living areas in South America, where they're embedded in a continuous forest canopy and they cut out an area to live. That's an anthropogenic perforation, but you can also get natural perforations created by very large trees that fall over. And because of the interconnectedness of the various vines and lianas between the trees and other trees, they will pull other trees down with them. And you can get a fairly large open patch exposed to a variety of different environmental conditions just as a consequence of this perforation. Now there's a catch here. We've known for many, many years that basically species diversity is maximized when area is large. So we tend to describe this in a species area relationship of large areas contain more species. The catch is, though, is that other ecological components in fragmentation sometimes do not have just a positive effect. Sometimes they have negative effects, which isn't surprising. I think we're quite aware of this. What's less well known, and what has sort of been at the heart of a lot of landscape ecology and a lot of debate in landscape ecology in the last 10 years, is how habitat configuration influences animal populations. And part of this is because we have no straight, simple rule for how all animals will respond to habitat fragmentation. And this is sometimes just a function of something as body size. A very small mouse lemur, a very small squirrel will perceive a fragmented forest in a very different way than something like a giant anteater, or something like a jaguar, or a gorilla, or a lemur. And so you can also get differences in the animal perception of the landscape based on whether they're arboreal or terrestrial, diurnal or nocturnal. And it begins to paint a picture of a lot of opportunities and a lot of fascinating ways of understanding forest fragmentation effects. Now, why should we care? Quite simply because no matter what we do, we are dealing with forest fragmentation. This is a graph produced in a recent paper looking at the effects of anthropogenic fragmentation on mammals throughout the world. Dark red areas are areas subject to extreme levels of fragmentation. Dark blue areas are areas subject to very low levels of fragmentation. And not surprisingly, arid areas, particularly like the Arabian Peninsula or parts of North Africa, as well as Australia, you see very little fragmentation because there is no habitat there to fragment. What we really want to focus on as tropical ecologists is the tropical region. And you can see much of sub-Saharan Africa is subject to massive amounts of forest fragmentation, as is the Indian subcontinent, Southeast Asia, and the southeast parts of South America. So these are expanding dominant features, and whether or not we can understand these is kind of the heart of the talk that I would like to have today. Now, it raises an interesting question. Is fragmentation a process or a pattern? And technically it's both. But what we've learned is that the great Lenore Frederik, who's sort of been at the heart of a lot of the debate about landscape ecology these days, what she's suggesting is that fragmentation is best studied as a pattern. In other words, the process isn't directly related to our models and our theories in landscape ecology. The process is one of landscape architecture, of use of scarce resources, of human habitation patterns, of conservation. What we really want to understand is the spatial configuration of habitat patches at one point in time. And then ideally, we want to compare these between landscapes. And what we're really trying to get at there is that we have these two elements of deforestation processes that we have to split apart. We have to understand habitat loss, but we also have to understand habitat fragmentation per se. So what is habitat fragmentation per se? This is where we're trying to control for the effects of habitat loss and center in on how spatial configurations of the remaining habitats are influencing animal responses to fragmentation. In effect, habitat fragmentation per se is about controlling for habitat loss and studying the fragmentation processes in and of themselves. Now, on the surface, that sounds fairly simple, but it becomes a little more complicated when you start thinking about how things can change. And the patterns by which fragmentation happens is fairly complicated. Just this very simple diagram showing you an original forest landscape, which is over on the left-hand side, with a number of patches of varying sizes and shapes. You can have any number of possible parameters that as a landscape ecologist, we want to measure and understand as a predictor value for how the animals are going to respond. Some of these patches in some of these landscapes now have reduced number of patches, some have increased isolation, some have reduced isolation, some have increases in average patch size, some have reductions in average patch size. So what it means is, it appears to our eyes that some patches get better and some patches get worse. But that's really a species-specific perspective. Again, the animal, the focal animal of our studies will determine how they perceive and respond to these various kinds of selective pressures. And here's the problem. It is a complicated process. The ecological effects of forest fragmentation are pervasive and wide-ranging and exist both on a spatial and temporal scale. You have the ultimate physical changes in what happens to the patch, but those in turn can influence demography. You can have changes in effect size, you can have genetic drift, you can have demographic stochasticity. In turn, the combination of both the physical changes and the demographic changes can result in community level differences. Now, the animals are not just sitting there not responding. They have their own selective responses. In fact, their own adaptive responses. They can shift niches, they can change their dispersal patterns, they can leave. They can just simply disappear from a patch. So it is complicated. There's a lot going on here. I'm an old football player. I like to keep things simple. And so if we try and keep it simple, I think it's a little more illustrative of the models and the way that we can use them to help us understand how things work in fragmented forests. Because really we have to get at the question of what aspects of this fragmentation process have a negative influence, a negative ecological response, versus those mechanisms that in turn have a positive effect on animal populations if we're interested in them. And this is along a continuum of increasing or decreasing patterns of habitat fragmentation. Sometimes the fragmentation is stochastic and we don't really know when or where it's going to happen, but with idealized selection using all kinds of remote sensing and satellite imagery, we can be highly selective in the landscapes that we want to look at. So today what I want to do, the overriding goal, is to understand a little bit about how fragmentation effects influence animal biogeography. And biogeography is an old science. It's basically just saying where are things and why, and where are things not and why. Now, I'm going to focus mostly on primates and lemurs in particular. It's what I know a little bit. As many of us know, lemurs are only found on the island of Madagascar, which is a large island, fourth largest island in the world, as my advisor always told me I had to use in every talk. Located off the southeast coast of Africa. And that shows you a satellite imagery of the island. The green indicates vegetation. The brownish grayish taupey color indicates different levels of disturbance of that vegetation. Now, why focus on lemurs? Well, lemurs are kind of a perfect taxon to pick for studying fragmentation effects because although Madagascar is large as far as island goes, at a continental level, it's quite small. And yet when you look, that little tiny island there, at the time of this publication, there was 103 species recognized by scientists. That number has gone up appreciably since then. That compares favorably with the 111 species that are found in Africa, and not that far from the number of species found in Southeast Asia. So you can have a lot of diversity in a small area. That helps, that's a larger sample size. From a conservation perspective, the lemurs that are found in Madagascar are probably the most threatened animals on the planet. 87% have threatened by various kinds of anthropogenic, largely disturbance, but also climate change, and all populations are declining. And so they are under threat and therefore expected to show very strong responses to various levels of habitat disturbance. The other thing is that there's just so many fragments on Madagascar. Madagascar has undergone massive forest conversion, largely as a result of climate change and human activities. When humans arrived on Madagascar many thousands of years ago, they began to exploit the environment, as humans do everywhere. And this has culminated now in a landscape of forest that is composed almost entirely of literally thousands of fragments of varying size. There is very little sort of continuous broad forest like what I was used to seeing in South America, where you could just walk for days and never reach the end of the forest. And now there's only a few places left in Madagascar where you had any hope of that. And unfortunately, what is left is burning. There is an increasing change in fire frequency that's occurring throughout the island. And it is principally located in western Madagascar, as well as along the remaining eastern humid forests. So that is contributing to conversion of forest habitats and contributing to further interest that we have in how animals are responding to this changing environment. Now, getting around Madagascar is always a treat. I had a colleague who went there once who said it was the only place he'd ever been where he traveled all day and got nowhere. And it is always remarkable to me that the resilience and diversity of transportation modes on Madagascar. It's just, you never know what you're going to bump into and you never know how you're going to move around. And it's always an absolute treat to me to travel across the country. When you finally do arrive at the forest, it is incredibly beautiful. I mean, just about everything you see in that forest is probably endemic to the island of Madagascar. And in fact, some of it may be only found in that region, and in many places of the island, that may be the only place that plant or that insect or that mammal lives. And so within there, you find these incredible endemic creatures, quite fascinating. Just about everywhere you go, it's an absolute treat as a biogeographer just to keep seeing all the amazing little creatures that run around in the forests of Madagascar. My favorite are the cryptic species, such as this is Europlatus fantasticus, which is the satanic leaf-tailed gecko. In the middle image, there's one that's pretty easily seen being hidden against the bark of the tree. Over on the right-hand side, there's one that's so well camouflaged that it's almost impossible to see, but there's the outline of it there in the tree. I'm always absolutely fascinated by seeing these creatures in the forest. And then ultimately, then we have the lemurs. I'm not supposed to say this, but they are incredibly cute. I'm supposed to say, as a cold hard scientist, that they're a fascinating study subject, which they are. But they always reminded me of stuffed animals that my youngest son always keeps up in his bedroom. So amazing, incredible animals. So the objectives today then, quite simply, are to look at three aspects of forest fragmentation effects. First one is edge effects, then we're going to discuss dispersal, and then we're going to look at conservation biogeography. So what are edge effects? Well, we'll start simply with defining what an edge habitat is. And an edge habitat is pretty simple to figure out. It's anywhere two different types of habitats meet. And so, for example, here, this is one of the edge habitats that we've been studying at. This is where grassland borders on a forest in northwest Madagascar in Ankarafantsika National Park. And down below, we have a different kind of edge. This is a highway that bisects the national park. And you can see in the foreground, there's a small mouse lemur there faced with the decision of traveling along parallel to the road or trying to cross it. And so the road itself represents a different type of habitat, creating a different kind of edge than what you see between the forest and the grassland. And so all of these edges have transitional abiotic and biotic characteristics. And these are things like wind speed, temperature, humidity, light levels, pollination systems, abundance levels, biodiversity, nutrient systems, carbon cycling. And it's stochastic, it's always changing. That's what makes it fascinating to me. And some of the characteristics of these edges could benefit or exclude certain species over time. So for example, no matter what you do in terms of forest loss, you're going to create more edge. It's just going to happen. As forest shrinks, as like this fragment patch shrinks, then edge becomes a disproportionately large part of the remaining patch. It also happens through habitat fragmentation. If you run a road through a fragment or you have a long, narrow fragment, it's going to be composed almost entirely of forest edges. So although these are independent processes, no matter what you do, you're going to get more edge. And so consequently, edges are probably one of the most dominant forest landscape features on the planet. And what you'll focus on here is the yellow and the orange. They both represent two different kinds of edges. So animals embedded in these patches then have to sort of decide on how they're going to respond to fragmentation. If they like the interior, they're core living species, then habitat fragmentation can actually result in the elimination of the species entirely from a forest patch. Whereas edge species may hold on, they like the edge, and they can continue to exist. So what I'm going to do now is describe one of the studies that we did looking at edge effects as it relates to genetics. And so we use population genetics to look at small-scale molecular edge effects in Microcebus ravalabensis, which you can see here in the image. And what we wanted to do was look at it to determine basically if there are any genetic differentiation between animals in the edge versus those in the core, because there was no data to support this idea, but we felt like it's something that should be able to be discerned in animal populations. And so we set up a study site in Ankarafantsika National Park, which is outlined in the hashed image here, and we moved along the edge down below. So we set up 3 transect lines for traps, one in the edge, one at 600 meters from the edge, and the one in the core of the forest. And that's one of my master's students there, Ryan Burke. We also participated with students from Dr. Radespiel's lab, and we sequenced a 529 base pair fragment of the mitochondrial DNA region. And to our great delight, we found considerable differences between the edge, the interior 600-meter area, and the core, where we assumed this was far away from any anthropogenic edge. In fact, the highest genetic diversity was found in the interior of the forest, with the lowest genetic diversity at the forest edge. And in fact, we never captured an individual lemur in the edge and in the core forest. And even though these weren't that far apart, they were within pretty close to the dispersal range of these animals. In many ways, they seemed like separate subpopulations, isolated from each other. And that was quite fascinating and quite surprising to us. So in other words, edge effects are an inevitable outcome of fragmentation and loss. And we know that edge effects can change abiotic conditions, and that can certainly have a massive impact on things like parasites, but it also influences biodiversity, abundance, and for the first time, the genetics of animal populations. So now we'll look at dispersal. Well, what do we know? Not that much. It's actually quite rare and serendipitous to capture dispersal. This is a periacephac, one of the rarest mammals in the world, who decided all of a sudden at one point to leap across 400 meters of open savanna to move between patches in Lemurs Special Reserve in northern Madagascar. We had no indication they were going to do this. They just decided to hop across the savanna. And surprisingly, we don't actually have a lot of direct information on dispersal distances. We actually have very few for fragmented landscapes. And just to show you how surprising this can be, these are two islands in the Florida Keys. And as a grad student, I worked on one of these islands on Raccoon Key. It was a monkey colony. And so every day I would go out on boats and we would study the monkeys. And one day we received a call from the sheriff on Big Torch Key, which is a human habitated island, that they had arrested one of our people and had them in handcuffs in the jail. We thought it was one of the technicians that got drunk and made a nuisance of themselves. So we went down to bail this person out, and we arrived in the jail cell. We discovered that in fact, what had been arrested was one of the macaques. The macaque had swam across 4.6 kilometers of open ocean to move from Raccoon Key to Big Torch Key. And this is shark-infested, high current areas, very difficult to swim in. So we were quite shocked. We brought him back, released him back onto Raccoon Key, and he did it again. He didn't know there were no female rhesus macaques on the other end of the island chain, but the drive to disperse was so strong. So how do you get at this? Well, one way to do this is to model this with metapopulation dynamics. And a metapopulation is just simply a population of populations that are connected by dispersal. There's all different kinds. There's patchy, there's mainland island, there's classical, and then ultimately there's something called non-equilibrium, which means that animals are patch isolates. They're never going to leave their home, as it were. And so what we did is that we wanted to do one of the first studies employing incidence function models to determine if lemurs have support metapopulations as a population structure. It's a lot of math. It's very complicated. Thank God for R. And basically what you're trying to do is derive at this logical equation that encompasses all these different kinds of metrics like colonization rates and extinction rates. And quite simply, you're trying to determine, do they or do they not form a metapopulation? And if so, what kind? And so we set up a research site called Abanja Bay, which is a fragmented forest site with 42 fragments. And this was largely done by my PhD student, former PhD student, Travis Steffens. We spent a long time walking in this landscape, looking at the lemurs that were there. And there were 6 of the 8 lemurs that are found in Ankarafantsika within this fragmented landscape. Going left to right from the top row, it's Microcephalus marinus and then Rebelabensis. And then the second row left to right is Carogales medius and Eulemur fulvus. And the bottom row left to right is Propithecus cockerelli and Lepilemur edwardsia. Well, results, it turns out that they do show metapopulation structure. In fact, the 2 small-bodied nocturnal mouse lemurs show strong evidence from mainland island metapopulation. And what's interesting is that Marinus also shows evidence for the among patch. And that's not surprising given their widespread geographic range throughout Madagascar. And Carogales medius and Eulemur also show an among patch. However, the Propithecus and the Lepilemur show no evidence of metapopulation structure. They are patch isolates. And so this shows us that there's a lot of data that can be collected by using metapopulation and modeling techniques. So fragmentation strongly influences dispersal and other aspects of life, such as colonization and extinction. And what we think our data are showing right now is it's really the large patches that are holding the key to species persistence in fragmented landscapes. Finally, I just want to summarize up things by looking at conservation by geography. Now, we've known now, and most people know, Madagascar has experienced extensive deforestation and overharvesting of trees and animals. And now they're faced with a new threat, which is climate change. And so we wanted to look at how we can model the effects of habitat loss and climate change on highly sensitive animal populations. And so we picked as our study species very sensitive seed dispersers that are diurnal, relatively large body frugivores in the genus Variesia. These are our two study species, Variesia variegata and Variesia rubra. They're critically endangered, and we wanted to see if we could determine what was going to happen in terms of their sensitivity to both habitat loss and climate change. And so what we did is we summarized approximately 88 years of data that were collected across literally thousands of research sites throughout the eastern humid forests of Madagascar. And really what we wanted to look at was sort of two ways of how deforestation was going to operate under climate change. One, with strict protection, where no deforestation was going to happen in protected areas, and another process in which we have relaxed protection, which means deforestation could occur in protected areas. And then we modeled this climatic and habitat-based niches of the rough lemurs along different representative concentration pathways. And these are just global emissions scenarios. The larger the number, the higher the CO2 emissions, the smaller the number, the lower the CO2 emissions. And here's what we found in terms of the actual rainforest cover under just climate change. Irrespective of the levels of CO2 increase, we're going to lose habitat suitability, which is the dark reddish brown areas. And we're going to lose, therefore, animal populations. Under strict protection, even with looking at climate change, we're still going to lose some very important habitat for rough lemurs, principally in the northern part of the island, but there are still some remnant areas that are going to survive in central and southeast Madagascar. With relaxed protection, we lose almost everything. If you allow deforestation in these protected areas, then all the areas outside of the protected areas lose their habitats, and you also lose the forests and the populations within the protected areas. And this is what this is going to look like, and basically through a time series graph. So on the left, we have relaxed forest protection. On the right, we have strict forest protection. So by the year 2080, everything in eastern Madagascar is gone. We've lost all the forests, we've lost all the animals, we've lost the critically important nutrient and water cycles that exist within these forests. Biodiversity is catastrophically destroyed on the island. So even with our understanding of current deforestation rates on the island, it looks like by 2080, we're going to lose almost 60% of the forests that are left on the island of Madagascar. And with climate change alone, we're going to lose about 75%. And if you combine the two of them with relaxed protection schemes, we will lose all of the forests on the island. So protecting protected areas is actually a very key conservation strategy. So we know that fragmentation effects are complex, but our increasingly complex models are proving to be very robust and informative. And really what we do in Madagascar sets the stage for how well we protect biodiversity in other countries, not yet subject to the same levels and intensities of deforestation and forest fragmentation. But it's important for all of us to realize is that it's really the decision of the Malagasy people to determine the fate of their habitat. We've exploited our resources here in Canada to no end, and we're only saved by the sheer size of our country. Our job, therefore, as foreign scientists working in a nation is simply to provide the best information to the people of Madagascar and allow them to make their own decision. Thank you. I'd just like to thank Dr. Radespiel for a kind invitation to participate in the seminar series. I always am completely thankful to the many, many guides and fantastic students I've worked with who make the entirety of my research program possible. And finally, to the various agencies that support my work directly or indirectly. I'm forever grateful. Thank you very much.
Thank you very much, Shawn, for this very nice and clear presentation. And we jump right ahead into the next presentation and keep all your questions, please, for the end. And the next speaker will be Frederik Kiene. And you can already, yes, you're already sharing your screen. So Frederik Kiene is a postdoc in my group, and he will present part of his PhD thesis that he finished just now back in April, and he will talk about the effects of habitat fragmentation on gastrointestinal parasites. And it's all yours, Fred. You may be muted. I'm not sure. I can't hear you.
Do you hear me now?
Ah,
yeah. Okay, now it's working. So thanks to Ute to invite me to this, to give me the opportunity to talk to you today and tell you something about our work on the gastrointestinal parasite infections, which we studied in small mammals in northwestern Madagascar in a fragmented landscape. And thanks to Shawn, who did a wonderful introduction into my into my work, and told you a lot about habitat fragmentation and about its impacts on biodiversity, which are enormous. And particularly like larger species, mammals, characteristic vertebrate species are studied, and many studies are showing effects or negative effects on the biodiversity of them. And you can see here in this scheme an ecosystem where you see that the different species are represented as balls, the consumer species in red and the producer species in green, and the trophic interactions are represented by the gray lines. But as you know, or as you heard from my title of this talk today, I want to talk about parasites, and those are not represented here in this scheme. But if we add parasites to a food web like that, it looks way more complicated. The parasites are now represented as the blue balls, and you can see that they are involved in a lot of trophic interactions, and they appear in quite high trophic levels. And the struggle is real. Parasites are very abundant, and it's estimated right now that nearly half of total biodiversity, so every second species, is a parasitic species. And parasites in general are very biodiverse. And parasites, as you can imagine now, that if they are so abundant and they're so numerous and ubiquitous, they might have important roles in ecological systems. And that's true because they affect trophic interactions massively, like, for example, predation and herbivory, but also themselves. Like here, some parasitic species you see, they appear in lower trophic levels, so they can serve as a highly productive nutrient resource themselves. And in this way, they are serving as a factor of stability for the population for population of the host species. And therefore, they might enable the coexistence of species and at the end produce a higher rate of biodiversity in the habitat. A good example to illustrate this fact is Plasmodium azurophyllum, which is a protozoan blood parasite, which is parasitizing two anole lizard species living on Caribbean islands. And we have a dominant, we have a subdominant species, and the more dominant one is more susceptible to infections with this protozoa. And this allows the more subdominant species to coexist in the habitat. So if we want to now look at parasite communities and parasites as parts of ecosystems in a now anthropogenically modified world, we don't know much about that because most studies, as I said, are focusing on the more characteristic vertebrate species, but also plants even sometimes invertebrate, but they are all free living species. We don't know much about parasites. So what we try to find out is which factors are making the differences in terms of responses of parasite communities on habitat alterations like fragmentation, habitat loss, and habitat modification, which could be differences in habitat degradation, for example, as well. And Shawn also talked already about a lot about Madagascar, and he already talked about one of our study sites, which we visited to do our study, and this is Ambanjabe in the Ankarafantsika National Park. So I don't have to say much about that. but we went also to a second site, which is the Mariano classified forest, which is a little part or a little bit further north, close to the ocean. And what we did there was organizing or installing forest transects from going from the forest edge into the interior of 40 forest fragments and 4 larger continuous forest patches, which are the light gray ones, and the forest fragments are the darker ones. And there we installed 61 transects. And along these transects, we captured 4 different species of hosts, model hosts, which we investigated for their gastrointestinal parasite infections. These were 2 primate species, the gray and the golden brown mouse lemur, and 2 rodent species, the endemic western tuft-tailed rat and the invasive black rat. And in fecal samples, in more than 1,400 fecal samples from more than 900 hosts, we performed coproscopy, which is a combined flotation sedimentation method, and looked for their gastrointestinal parasite propagules. And these 16 different morphotypes were what we found in these samples. But in order to now try to find out something about parasite ecology in dependence on habitat fragmentation and habitat degradation parameters, we had to smaller our subsample because we could just study morphotypes which were quite abundant. We had a threshold of 5% here, so the prevalence of the morphotype had to be higher than 5%. And so we were left with these 5 morphotypes, which we studied and modeled in the further process. And we used a generalized linear mixed modeling approach to do that, and used the presence-absence data of these 5 parasite types and the parasite species richness, which is the number of simultaneously present parasite types, but now all 16 different parasite types are included. And we use these as dependent variables in these models under the control of random factors, which were the location, so Ankarafantsika or Mariano, and the sampling year, because we sampled our hosts in 2 different years, in the years 2017 and 2018. And what we wanted to test were the effects of different sets of fixed factors. And these were host-related fixed factors like host species, host density, host sex and body condition, but also habitat-related factors. And an interaction term, which is controlling for different habitat preferences of the different host species. Due to the limited time, I want to focus today on the habitat-related factors, and I want to introduce them here. And these were factors or like effects of fragmentation, which we tried to test by comparing continuous forest sites with sites in forest fragments. But also we tested the impacts of forest area. We tested effects of edge effects by using these 2 factors, which were the distance of the capture place of the hosts here along the transects to the closest forest edge, and the percentage of edge habitat of a fragment, which was defined as habitat which is closer than 50 meters to the closest forest edge. I can't proceed. Why? That's just slow. And habitat degradation was also tested with 2 factors which we used, which are 2 gradients. One called vegetation clearance, which is a gradient from rather dense vegetation to more open areas, and forest maturation, which is a gradient from secondary to primary forest. And these factors were derived from a principal component analysis, which is an analysis which is generally used to condense complex sets of data to fewer principal components with high content of information. And we used as a base for this principal component analysis the data from Bertrand, Bertrand Andrea Tituaina, who did a lot of vegetation plots in all our transects. And we took average values of like vegetation parameters, like the numbers of saplings, numbers of trees, numbers of lianas, but also the number of zebu scat, number of cut trees as signs of human presence. And we took average numbers per 100-meter part of a transect to insert into the principal component analysis. And we ended up with the 2 most significant principal components, principal component 1 and 2. My computer is, delayed or like, yeah. Do you see any? Sorry. Yeah, we ended up with these two factors, forest maturation and vegetation clearance. Now to the results. I tried to illustrate them in this table. And here you can see that we found significant differences between the forest, with the continuous forest and the forest fragments for the Strongyloides nematodes. And also for the Strongyloides nematodes, there was a significant positive effect of forest area. So prevalences were higher in larger fragments or larger forests compared to smaller ones. A very nice result is that the proximity to the edge was showing to be very negative for the presence or the prevalence of the parasite morphotype studied. You can see this here in the scheme or in this mosaic plot, where we have the two affected parasite morphotypes and the gastrointestinal parasite species richness. And you see here that we have foes coming from the forest edge on a gradient going into the forest interior. And you see here that the prevalences of these two morphotypes were much lower at the forest edge compared to the interior, and also the total parasite species richness, so parasite diversity, was very low, or was lower at the forest edge compared to the forest interior. Going further on, the factor vegetation clearance had an entirely negative effect on the parasite types studied. So we found lower prevalences here and more open areas for these parasite morphotypes, and in total for the gastrointestinal parasite species richness. If we now look at the overall picture again, we can see that it seems to be that there are two different groups showing two different kinds of responses for parasite for habitat fragmentation. There's this first group here, Lemuricula and Enterobine, which didn't show much of a response on the environmental factors here. This is probably related to, or most likely related to, the biology of these different types of parasites, which is different for these two morphotypes to these three morphotypes. It is so that the Lemuricula and Enterobine nematodes are directly transmitted parasites. They are pinworms, and they are transmitted in the direct contact from host to host, and they don't spend any time in the environment for development because they need to be uptaken right after being shed with the feces of the previous host. So we can say that these two morphotypes are showing a quite high level of host association. If we now look at these three parasite morphotypes where we found a lot of significant responses to the investigated factors, we can see that the Strongyloides morphotypes, the Strongyloides nematodes, spent very long time in their life cycle in the environment, either in the during development before they parasitize in successor hosts, but they can also perform full environmental generations, which has a lot of opportunities for the environmental factors to influence parasite dynamics. And these two morphotypes, Suboluridea nematodes and Spirulid nematodes, they are both known to be heteroxenous parasites, which means that they need arthropod intermediate hosts to be infectious again for successor hosts. And they spend in between long times in the environment for development, for parasite development. So we can say that these three parasite morphotypes, which are under high influence of the environmental factors, are showing a quite high proportion of the whole parasite population in the environment compared to the pinworms. What we can say at the end is that the factor vegetation clearance seemed to be a central factor in our study, because I think that it's very likely that all the effects of the other factors are related to the different vegetation conditions in, for example, edge habitats compared to interior habitats, and in small forest fragments compared to larger forest fragments. It's therefore my colleague Bertrand Andriatsutreiner could show in his study that the vegetation, for example, was significantly different at the edge compared to the interior. And it is very likely that, or it is clear that edge habitat is more pronounced in smaller forest fragments and in fragments compared to the continuous forests. So to conclude, we can say that habitat fragmentation and edge effects are detrimental to parasite diversity, at least for the investigated habitats in the dry forests of northwestern Madagascar. And these negative effects are mediated by vegetation factors, or by the vegetation which is leading to suitable habitat conditions for the development of either parasites or their intermediate hosts. And this makes the nematodes with free-living generations and the nematodes requiring arthropod intermediate hosts particularly susceptible to habitat fragmentation and habitat degradation effects. Parasites with high levels of host association are in contrast, or could be shown to be not susceptible to, or so much susceptible to environmental changes. At the end, it's left to say that the most affected parasite species are very likely endemic habitat specialists, just occurring in northwestern Madagascar. And I think this, like looking at the whole world, our general picture, our results show that the general parasite diversity, which is mainly representing habitat specialists, is very threatened by this development. And by looking at the numerous services of parasites beneficial for ecological societies or ecological communities, it is a very alarming result. And I want to now thank you for your attention, and I'm very happy to hear your questions. And at the end, I want to encourage to read our paper, which we published on this study early in this year in Ecology and Evolution.
Thank you very much, Fred.
That was it. Thank you.
Yes, for this presentation, and I think it nicely adds up or complements the findings that, or the talk that Shawn has already given. And we're moving smoothly over to the third talk, which is still connected to fragmentation. And if you stop sharing, Fred,
yeah, I try.
Gabi can upload his presentation, which will be presented by Gabriele Sgarlata from the Instituto Gulbenkian de Ciencia in Oeiras in Portugal. And he will also talk, well, I'm not sure only about Madagascar, or maybe yes, we will see. Okay, Gabi, it's all yours.
Can you see my screen?
We see the presentation mode, but not the presenter mode, but not the.
Okay, I will try again.
Not the picture that is meant for us.
Now you should see.
This looks better, but not yet the yes, perfect.
Yeah, so yes, I'm Gabriele Sgarlata. I'm a PhD student at the Instituto Gulbenkian de Ciencia in Portugal, Oeiras. And I am a PhD student at the Population and Conservation Genetics group under the supervision of Lunes Shiki. So today I will talk about a part of my PhD work on what we understand in terms of population genetics concerning habitat loss and fragmentation. So how habitat loss and fragmentation can theoretically influence genetic diversity and genetic pattern from wild populations of animals or plants. Part of my PhD, I worked with lemurs and rodents in Madagascar, but today I will actually exclusively talk about my theoretical work relation to what we can understand in terms of the population genetics and habitat loss and fragmentation. So as already shown, as introduced, the question of forest loss and fragmentation is quite strong, in the sense that today, thanks to satellite images, we have a more quantitative understanding of the extent to which natural habitats are fragmented. And this work of 2015 has shown that indeed nearly more than 50% of the forest today worldwide are within 500 meters of an edge. So here in this picture, you can see in blue the forests that are continuous, and in red, orange are the forests with that are severely fragmented. Now, As Shawn has said, and I will actually pass quickly through it, the process of, or at least the phenomenon of habitat loss and fragmentation should be, and we need to look at it at different spatial scales, because the mechanisms or that act may be different. And in particular, I'm talking about landscape scale. So the phenomenon that Shawn mentioned as a breaking apart of a continuous habitat, for instance, a forest in patches, which you see here as a green square, or habitat contraction. So will be basically a mixture of different processes, which are habitat loss, the fact that you have reduction in habitat amount, increase of edge effect, because the fact that you decrease habitat amount will expose individuals or species that are more internal to the habitat to a different type of ecosystem, let's say, and then habitat isolation. So the state of the art concerning this topic, it actually started, let's say, in the 60s, when this question was started to be addressed. So what is the impact of habitat loss and fragmentation on biodiversity? This was work done in UK. And since then, the question of course, has been addressed by different teams on different perspectives from an ecological point of view, mostly, and more recently also population genetics, so looking at genetic diversity. But what is now a bit, what it can appear by reading in the literature is that although this question is relatively old, there are still some controversy. And I want to expose this controversy by looking specific to one, let's say, example, where basically in 2017, there was this important paper by Frederik, which showed that fragmentation per se, by doing a meta-analysis, showed that fragmentation per se may actually be positive for some species, for most species that were analyzed in that work. After this work in 2017, there were several responses because it was kind of counterintuitive and was putting in discussion the major view of habitat loss and fragmentation as a negative component for biodiversity. So from one side, we had indeed a response of a series of other scientists that actually brought evidence for from a series of experimental studies, controlled experimental studies, which were actually showing that factors such as habitat size, patch size, or isolation and edge effect indeed actually showed a negative effect on biodiversity, which is showed here in these panels by these black dots on the left side. So when the black dots are on the left side, it means that there is a negative effect. When they are on the right side, it means that there is a positive effect. In all of these cases, there was a negative effect. On the other side, in 2020, there was a paper which also Frederik was author, which was basically a meta-analysis of different taxa, across different taxa, of the effect of habitat amount, isolation, patch size on a species diversity, so species abundance. This study was showing that basically there was a lot of effect of landscape scale processes and not much of a local scale. Although it could be seen, for instance, in the case of patch size, that we had some significant effect, I would say, on species abundance. Therefore, one thing that, let's say, these two perspectives agree on, one in which claim that fragmentation per se is not really important for species diversity, and the other side, that another perspective that states that actually it is. In both cases, they both agree that habitat loss is negative for biodiversity. But so this is the ecological point of view, but what about genetics? So only recently, with the help of DNA sequencing, the decreasing cost of sequencing technology, now it has been possible to gather much more data across different taxa across the world. And I would say that in the last 10 years, we had a much, let's say, clearer understanding of the extent to which habitat loss and fragmentation influence genetic diversity. What I'm showing here is just one paper of last year, yes, 2019, which they compare different taxa and different variables indicative of habitat loss and fragmentation. And they also confirm that even for genetic diversity, habitat loss fragmentation has a negative influence. So why talking about genetics? It might not be obvious. And the reason why is that, as I said, so trying to understand genetics and genes is a way to, in a way, it's a proxy of amount of individuals in the habitat. Therefore, could be useful to understand the state of endangered of a given species. But it's also important for possible adaptation in case of climate change or environmental change in general. But it's also useful, or we are learning much more on how to use the information encoded in DNA to reconstruct the past. So here I'm, for instance, showing a recent review which they basically studying the genome of humans, population across the world. We are little by little drawing a story that help us to understand where humans came from and how did they move across the world, and how did they colonize the world. And finally, our understanding, theoretical understanding of genetics and the mechanism of inheritance is fundamental to then be able to predict what is going to happen to species at the level of genetics diversity once habitat will change. So, as I said, I work both on theoretical approaches, try to understand genes and population genetics in space and time, but I also worked in Madagascar, sampling DNA from lemurs and rodents. And what I'm trying to highlight in this slide is that I think these both type of work are fundamental because as Shawn was explaining earlier, there is this dichotomy between pattern and process. I will say that from one side, the empirical approach, which helps us to see what we can detect today from genes, needs to a way help to be interpreted with the use of analytical, for instance, or simulation models. So theoretical approaches that can help us to understand how we get to a given pattern. So I will show my results of this study that I've performed in part using spatially explicit simulation, which means I'm basically simulating habitat in space and time where there are species living in. And one major characteristics of those simulations is that individuals are geographically limited in dispersal, which means that they will tend to reproduce only with individuals that are geographically close and less likely with individuals that are far apart. In population genetics, this pattern can be visualized usually by correlating geographical distance with genetic distance. So if you take samples from a wild animal or a plant, and you correlate geographical distance with genetic distance, and you observe a positive relationship, this is usually indicative of the fact that in that species, individuals are geographically limited in dispersal. So they cannot go far away to reproduce, but will tend to reproduce more closely distance. I'm showing some simulation that I did to really understand the effect of habitat contraction, distribution of genetic diversity. And to do that, I looked at two processes. One is habitat loss, so yes, loss of habitat, and edge effect. And I asked, how these two factors influence the distribution of diversity within the habitat patch. So I simulated several replicates for thousands of generations. I took, I'm showing here the simulated scenario. It's a very simple square. And I took around 30 genetic markers, and I sampled 14 individuals along the diagonal of the square habitat. And I used heterozygosity, observed and expected, and mean number of alleles as measures of genetic diversity. So here, I'm showing some of the results. So you see that in the x-axis, you have the position along the diagonal that you can see here in the scenario I have simulated. And the y-axis, you have the difference in diversity between the population or the individuals that I have sampled in the center of the habitat and the individuals that I've sampled on the corner. And you can see that basically by simulating a different dispersal rate, what is not obvious, at least for those that work in population genetics, is the fact that even at high dispersal rate, so the purple line that you can see here, you actually observe a significant difference between central population and corner populations. So these initial observations show that really there is an effect of habitat edges following the assumptions of the model that we have simulated on genetic diversity. And this is even more striking if we compare the loss of genetic diversity with the absolute value of genetic diversity at the center of the habitat. That is, I'm showing here in the x-axis the level of dispersal rate, and the y-axis the loss of a percentage of loss in genetic diversity. So across all the measures that I took and across all the dispersal rate, you can see that the minimum amount of genetic diversity that we lose is about 5%, and the maximum is at 30%, saying that really habitat edge effect can have an influence, a significant influence on the amount of genetic diversity of a given species. However, we use more complicated models, so the mechanism of inheritance of genes, to try to understand why edge effect influence negatively genetic diversity. And to do that, we use what I call the coalescence theory. So basically, the idea is that you take different samples, so different individuals in the habitat, and by comparing the amount of genetic differences between individuals, you can say, okay, this individual had a common ancestor in this location in space and at the given time, that you can estimate using genetic data and models. So what we hypothesize and what we think is that basically on the populations that are on the edges, what happens is that individuals, given that they cannot disperse across different directions and do not receive genes from many directions because they are constrained in that habitat edges, they will have a higher rate of relatedness among individuals, which then translate in a lower genetic diversity. So what I did was to try then to go for a more complex scenario, because of course, habitats in reality are not square, but are more of a complex geometry with multiple edge effect and spatial habitat heterogeneity. So what I tried to do was to develop an approach which merged landscape ecology and population genetics to have a measure of edge effect, which can account for complex geometry and habitat heterogeneity. I use this classical approach in landscape ecology, which is called focal processing, makes an average of the habitat amount, considering also the edges. And from these, I combine this information with classical spatial population genetic models. So, what you can see is that if we assume what I call an equilibrium population, I'm here showing the correlation between the measured genetic diversity across the simulation that you can see as colored dot, And on black, you have the analytical solution of the model that I basically developed, standard, linking landscape ecology and population genetics. You can see that basically the black dots are in very close proximity with the simulations that are performed, suggesting that this approach could be used to test habitat edge effect and to have a more fine scale estimation of what will be the level of genetic diversity due to complex geometry of the habitat and habitat heterogeneity. However, in reality, we have cases in which habitat have been changing several times across time. They may not have necessarily reached a form of equilibrium of the system. So what I've done, I also extended this model to be able to predict what will be the dynamic of the change in genetic diversity once you have a contraction, by taking into account both habitat loss and the changes in edge effect. And again, here I'm just showing the results where basically you see in the y-axis, in the x-axis, we have time. The dashed line is the time of the contraction, of habitat contraction. And the y-axis, we have the level of genetic diversity. The dot represent the simulation results, and the line are the prediction from the analytical model. You can see that by taking populations in the corner or in the middle or in the edge of the habitat, and by simulating different levels of dispersal rate, the analytical model can well predict or follow the results simulated, suggesting that we can, in principle, use this approach to really have some prediction about the future. So what's going to happen, level of genetic diversity after contraction, but also to reconstruct the past. I think I have to conclude because I'm over time. But I will try to pass very fast on another aspect of my project, which is to look at the landscape scale. We are interested to really understand if we take populations on different forest fragments and we compare genetic differences among them by considering space and time, can we estimate when deforestation has occurred in a given landscape? To do that, we look at an isolation by distance pattern, so this correlation between geographical distance and genetic distance. What we ask is how this correlation change once the habitat is fragmented. Because a characteristics of this correlation is that the habitat has to be continuous and not fragmented. So it's still unclear in the community how would change this pattern once the landscape gets fragmented. And this research was motivated by the fact that in real landscape, you often see that even if you sample different forest fragments that are in principle isolated from among each other, you often see significant IVD. We cannot explain why, but with my work, we have tried to explain it. And yeah, so basically I performed some simulation which showed that indeed that dispersal rate has an effect on for how long we can detect this pattern. But what I wanted to show, because I'm late, is that we have developed a model that actually is able to, by using some mathematical simplification, to have an estimation of how genetic diversity change in space and over time once you have deforestation, so fragmentation of the habitat in habitat fragments. And here I'm showing the case of low dispersal rate, and here are high dispersal rate. What my results have shown is also that not only dispersal rate has an effect, as you can see on the fact that here the different colors are population at different distance, geographical distance. And you can see here that are more clear, the differences between the different colors, whereas here are more compacted. But we also observed that the extent of the fragmentation, so how big are the fragments that you generate after habitat loss fragmentation, can be very impactful on what is the dynamic of the genetic diversity over time. You can see it by comparing this plot with this plot. So I think I will conclude here. by saying that, yes, so go back to the initial slide. So genes can help to understand genetic diversity today, what we observe today. Understanding the rules of inheritance of genes may help us to reconstruct the past, but also to look in a way to the future. And I thank the people of my group and you for the invitation. And yeah, that's it.
Thank you very much, Gabi.